I got the fever. I'm heated, pumping up the meter. Roll me a ringleader, mad party bleeder. Make this with a storm, lead after lead after leader. Hey, can you dig it? I come alive, calling, give me what you got. Back up, stick up, pump your fist up. You check the side of the buck shots, a bus slip up. Till the morning like a kid, gonna make you feel alright. Pump it up all night, body rock, show me what you got. Let Rob break the moves. Till the morning like a kid, gonna make you feel alright. Pump it up all night, body rock, show me what you got. Let Rob break the moves.
Do you wanna get
Check it out, check it out, check it, 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 check it out.
I gotta beat this guy in 2 minutes. Good morning, everybody. I'm trying to finish this game. I shouldn't have started it. I'm glad you're vibing, Connor. This guy's in trouble. This guy's in big trouble. Big, big trouble. Hmm. Hmm. Interesting. Hmm. That's an interesting move. What was that for? Huh. Ooh, interesting move. Interesting move. Alright. You are under attack, sir. What will you do? You're dead there. Dead there. Not dead there. Okay. What are you gonna do now? Alright, let me see. Let me think. Rook. He's gonna die there. King is guarding. Can go here. Guarded. That's guarded. That's guarded. That's guarded. Okay. Trap. Goodbye, queen. Evil queen. You will be smited. Let's see. Everywhere he goes, he's dead. Except for... No? Dead? Dead. 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 Definitely dead. 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 That is a trapped queen, ladies and gentlemen. I don't think that was a wise decision. Okay. Just give me that, please. Thank you. Okay. Double pawns. Yeah, that's pretty free right there. But I think a better move is right here. I think this is a good move. Okay, he captures. That means I'll capture here. Yeah. Capture's good. Okay. I expected this. Let's just move our pawn up. Sorry guys, this game really took longer than I expected. I should have played like a 3-minute game. We'll get class started, I promise. Oops, that was my move.
Tell you you're splendid. You tell me I'm wrong. The smile on your face and your blue eyes make me go to space, far away.
Checker. I could figure this one out. Okay. Die! Just lose the game. You lost. You're done. Dunzo. Okay. That's a weird move. You just lost your rook. Come on, guy. What were you thinking? What were you thinking? Okay. Chaku. What do I do here? This is a good one. Okay. Pop, pop, pop. Goodbye. Alright, let's get started. You want a rematch? No. No, I'm sorry. No rematch. You don't get to have a rematch. Alright, good morning. Good morning, gentlemen. Make sure that we are live. We are live. Of course we're live. Good morning, Miss McGee. Alright, let's see who's here. Christian is here. Bryce is here. Connor, Adam. And that's it so far. Alright, well, let's get started. Today we're going to talk about predicting certain traits that will appear in subsequent generations. So let's open up my stuff here. There we go. Man, it is warming up. Has anyone noticed that the weather for the next few days is like in the 90s? Also, Mr. Holden, 10 o'clock. I gotta go for a doctor's appointment. That's fine. We should get this done before 10. Easy peasy. Promise. Alright. We are at 11.2. Applying Mendel's principles. And in order to apply Mendel's principles, we have to have an understanding of probability. Now, what we know so far about genetics is a few things, okay? We are aware that there are dominant and recessive alleles based on the principle of dominance, right? There are alleles, or quote-unquote versions of genes that are dominant over other versions of that gene. And if you were to have a gene combination that included a dominant allele, the trait of that dominant allele will be the one expressed. So, for instance, we have a look right here. Whenever we crossbreed plants with round and wrinkled seeds, the offspring end up with all round seeds. That means that round has to be dominant. Yellow and green seeds, when those traits are bred together, the yellow seeded trait is what appears in the offspring. That means that yellow seeds are dominant to green seeds. And the seed coat, gray versus white, when you crossbreed individuals that have those two different seed coat colors, the offspring all have gray seed coats. So, again, gray is dominant. All right, there are traits that are dominant over others. That means they'll be expressed. Does that mean that the recessive or the non-dominant allele is gone forever? No. Okay. We know that it's possible for recessive traits to skip generations, and we see that right here when we include the original parent cross, the P cross, or the P generation, and we look at the F1 generation. It appears that we've lost this particular trait. It was dominated by the tall allele. Now, when we breed those individuals, they do have those short alleles in their genes, and it's possible to pass on that short allele and result in short offspring in the F2 generation. Okay, the F2 generation. Why does this occur? Well, Mendel understood that there were always a pair of alleles for every single trait, at least two alleles for every single trait expressed in an organism, and those specific alleles could separate individually when sex cells were formed so that they could be recombined into new combinations that were not possible in the previous generation. So that's why the shortness trait skipped the F1 generation and then later appeared in F2 because these although these particular gene combinations, the dominant T and recessive T, dominant T recessive T, even though those will express tallness in the plant, it's possible to separate those recessive alleles through gamete formation and then recombine them to have an individual with two recessive alleles as their gene combination. Okay? So that's where we're at so far. Uh, we understand principles of dominance. We understand how alleles separate and then recombine during gamete formation and reproduction to produce new combinations that were not possible in subsequent generations. Okay? So basically what I'm saying is no matter how many times you do it, right? If you take a tall plant with two dominant tall genes, right? Remember our dominant T is the tallness trait, or the gene for tallness, and the lowercase t is the gene for shortness. And the reason why we're sure of this is because remember, this P generation, they were the true breeding plant stock that Mendel kept. Every single one of those individuals came out tall or short or whatever trait that Mendel was controlling. He made sure that it was only that particular trait and only that particular combination of alleles. And he would breed them over and over again. And he would only get tall plants, and he would breed the short true breeding stock over and over again, and he would only get short plants. So he knew the gene combinations for these two particular parents in the P generation. Okay, it is only possible to acquire a genotype with a dominant and recessive allele in the F1 generation. It's not possible to have a plant that is short in the offspring of this particular generation. Okay? It's only possible once we get to F2. Alrighty, so there's a couple things we need to break down before we continue. We know what alleles are. We know what genes are. We know how to represent alleles using just simple capital and lowercase letters. But let's kind of step back and break things down a little further so we have a really clear understanding of what these gene combinations mean, how to address them, how to define them. So let's talk about this for a second. We need to recap because it gets confusing when I'm throwing around the word gene and alleles and trait and genotype and phenotype and all this stuff might get you mixed up. So let's sort of outline a few things right here. So we're aware that genes, right? Genes are they're basically sequences of DNA that code for specific traits, right? Right? Let's go ahead and just say, well, let's take your book's definition actually. I don't want to mix you up and have it appear differently on the test. Let's pick the book's definition. All right. A factor that is passed from parent to offspring. Okay, they use that. I mean, at the same time, it does control traits. But these are factors passed from parents to offspring. All right? The next thing we need to be aware of is what an allele is. And I remember my 4th period class last year loved to say allele for some reason. I think that was when the Pokemon Sun and Moon was still around, or Pokemon Sun Ultimate, or Moon whatever. And people were still like all in that Alolan phase, so they were saying allele. Haha, interesting. Why do I have Rockstar games on my computer? Because I'm a consumer and I can purchase what I want to in this free enterprise economy that we live in. That's why, Adam. So alleles are simply different versions of genes. Okay. For example, you can have 2 different versions of the plant height gene. You could have tall or short, right? So when I say allele, I'm talking about a version of a gene, right? I'm talking about a version of a gene. And alleles, along with genes, are passed down from parents to offspring. Each parent contributes one allele. Let's go ahead and uh, address this right now. Because of segregation, alleles separate during gamete formation and later recombine during reproduction to form offspring gene combinations. Let's go ahead and stretch this out. This is sort of, we're kind of breaking down what we should be aware of. Remember, alleles separate during the gamete formation. They recombine during reproduction to form offspring gene combinations, okay? Uh, this is important. I mean, I'm not going to write down that we understand we should already understand the principle of dominance, okay? So I'm not going to write that down. But we need to expand upon what these gene combinations are, okay? So, the term I'm going to use when I'm referring to a combination of genes is genotype, okay? Genotype is simply a combination of either recessive or dominant alleles. Let's find your book's definition. Uh, book's definition is right down here. Genotype. Genotype, they define it as the genetic makeup of an organism. I like to call it just the combination of alleles. An organism's combination of alleles. And there's, depending on whether the trait itself is controlled by multiple genes or a single gene, it could be two alleles that make up that combination, or several dozen sometimes. An organism's combination of alleles. So, for example, the ones we saw in our lessons on Tuesday, we saw this particular combination, the uppercase T, or dominant T. We saw dominant T, recessive T, and we saw recessive T, so two lowercase t's. Okay? As we could see, with two alleles that control a specific trait, there's only three possible combinations. Two dominants, a dominant recessive, or two recessives. And that's where we're at right so far. This is different from phenotype, which is also introduced in your chapter. Phenotype is distinct from genotype. Now, they are related in the sense that the genotype controls the phenotype, but the phenotype is quite well, it's simpler. It is simply the expressed trait in an organism. Okay. Whatever that particular organism displays or expresses as their trait is their phenotype, right? So, an example of our pea plants, the two possible versions of the trait that they can express are tall or short. And we see in some organisms and other organisms that combinations of alleles will give more than two phenotypes, essentially. Examples are the rabbit, the color of a rabbit's fur coat. Another example is human eyes and human skin color. There's more than just two phenotypes. But in pea plants, because it's a trait, tallness is a trait controlled by only two alleles, we see very few, little variety in the traits of our pea plants. Okay? Um, so that's all I'm going to update you on right now, but we should be aware of all of these things, okay? Again, genes are factors that are passed from parents to offspring. Alleles are different versions of genes, examples being there are alleles that might express different plant heights in pea plants. So the example we have is tall or short. And it's possible for alleles to separate and then recombine during gamete production and then later actual sexual reproduction, and we're able to form new gene combinations, which we call a genotype. Any combination of alleles is a genotype. And a phenotype is whatever the expressed trait is in an organism. Okay? Let's talk a little bit more about genotype. So there are a couple things we need to discuss in this area. There are different terms for different combinations of genes. And the two that your book really highlights are homozygous and heterozygous. Okay? These are quite simple. All right? what does homozygous mean? Well, homozygous simply means that uh, whatever particular genotype this organism has, it has two of the same allele. Is a genotype uh, where the organism has two identical alleles. And for the example of our pea plants, the only two options are either two dominant Ts, two dominant alleles, or two recessive short alleles. Those are the only possibilities in our pea plant example. Okay? And we have heterozygous. Heterozygous is a genotype where the organism has different alleles. They're not the same allele. So, let's write that down. And the only possible combination in our pea plants is the dominant T, recessive T. And we refer to, when it comes to homozygous alleles, we refer to, well, let me get rid of this. We refer to our different, either, when we have two dominant alleles in a homozygous genotype, we call this homozygous dominant. So we say dominant here. And when we have two recessive alleles in a homozygous genotype, we call this homozygous recessive. Um, but you know, if you'd rather not use the terminology, I'm okay with people saying, oh, we could do capital T, lowercase t, or dominant T, recessive T. But it makes me so happy when my students start using these terms. Oh, Mr. Holden, this blah 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 blah. This can't be possible because this individual's homozygous recessive. And I'm just like, oh, my heart melts with joy when people use the terms that I introduce to them. But uh, an allele is a version of a gene. It is not. It is not necessarily the whole gene. It's just a version of it. Okay. So there we know the names of our genotypes. We have homozygous dominant that has two dominant alleles. We have homozygous recessive, which is two recessive alleles, and we have heterozygous, which is a dominant and a recessive allele. Okay, so those are the terms we're going to use moving forward. All right, and we'll touch upon phenotype later. But the next step from this point is to uh, well, we have to engage a little bit of mathematics in biology now. So forgive me. I know this is not a math class, but we're going to jump right in and talk about probability. Let me scroll up here. So probability is essentially the likelihood of particular events happening, a probability of it raining, a probability of a car crashing into you on a given day, probability of getting a cold. I mean, there's always a likelihood that a particular event will happen. And it's not always possible to calculate the exact probability, um, but in the case of genetics, it is. We can calculate an exact probability of particular events happening. Or in this case, those events are particular traits and genotypes that will appear in subsequent generations. All right, that can be calculated. But before we do that, let's just briefly do a little exercise on probability right now. So the book likes to use a coin flip as an example of a way to understand simple probability, and I agree. The probability. So you guys tell me the probability of flipping a coin and getting heads. Go ahead, just type it in the chat. Tell me the probability of flipping a coin and getting heads. It's right there in front of you guys. Check it out. You guys and your pufferfish. Alright, if you were like, Mr. Holden, why would you ask such a redundant question? Obviously, the probability of getting a heads on a coin flip is simply 1 out of every 2 chances. Oh, you know what? I should probably do this on a paint pad. I'll do it on a paint pad. Can I make this larger? I can't. Alright, new paint pad. So the probability of getting heads on any given coin flip again, is 1 over 2, or 50%. And we're going to assume that the coin is not a trick coin, it is not a cheat coin or anything like that. So any particular coin flip, the chance of you getting heads is 1 out of every 2 flips. That is the true probability. Those are the only 2 outcomes to a coin flip. So we should know that our probability is a flat 50%. Okay? Flat 50%. And we can also calculate this further, like what's the possibility of getting heads and then a head? So what if we get a heads and then another heads in a row? What's the chance of that happening? We flip the coin once, get heads, and flip the coin again and get heads. Well, all I have to do is take our 1 over 2 probability and multiply it by another 1 over 2 probability, okay? And we would get the chance of you flipping 2 heads in a row is 1 over 4, or 25%. Okay? And if we could do this even more if we wanted to, we could keep expanding this further. What if we wanted to get 3 heads in a row? Well, again, 1, 2, 3 heads in a row, we'd have to multiply the probability of each individual coin flip together. And we get the chance of getting 3 heads in a row being 1 out of every 8 tries. Pretty steep. But what if this happens? What if you actually flip a coin, right? And you end up getting heads 3 times in a row. Alright? What does that mean? What does that mean if you get heads 3 times in a row? Go ahead and type what you think that would mean. Or what if you got 5 heads in a row? How about that? Tell me what do you think that means? We're not assuming the coin is rigged. We're assuming the coin is just a regular coin, just like any other coin. It's not a cheat coin or rigged coin. The game's not rigged. It's just a regular old coin. I think Bryce hit the nail right on the head. You got lucky. You met that improbable curve of a finite probability. You just need to flip it. You just need to flip it more times. Okay. You might look at a coin. Let's say you get 5. You flip a coin 5 times and you get 5 heads in a row. Okay. Do you need a new coin? No. Okay. You might look at this and say, okay, there is basically a 5 out of 5 chance that I will get heads because I flipped it 5 times, and each of those times it came out heads. So this is a 100% chance for a get landing the coin flip landing on heads, right? You might assume that, or you might assume that it's just really high and it favors heads more. But I'm going to counter you with my favorite mathematical statement, uh, and I pretty much live by this phrase. I don't want to write the actual equation out because I don't know it, but I can write sort of a loose math English version of it. Okay. All right. X bar approaches mu as n increases. What the heck does this mean? Okay. So let's go ahead and just address n really quickly. What the heck does n mean, Mr. Holden? N is simply sample size, right? And in this case, our sample size is however many times we flip the coin. So in this case, we flipped it 5 times, okay? That's what we got here. We flipped the coin 5 times, and we got 5 heads in a row, all right? That is our n. This is our sample size, okay? What is x bar? What the heck is x bar right here? Well, x bar is simply the sample average, all right? Or the sample mean. I'm going to say mean here. Sample mean, sample average, both mean the same thing. And what is our sample mean in this situation? Our sample mean is here. This is the average that we got in our 5 coin flips. We got 5 out of 5 heads, and that's 100% average for heads. Okay, and that just leaves us with mu. What is mu? What is this weird thing right here? And in case you're ever wondering what a mu is, it's just simply a u with an extra tail that extends out longer like that. We call that Greek letter mu. Mu. All right. So what is mu? Mu is the true average, also referred to as the population average. But in this case, I think it's better to understand if we say it's the true average. Oop, I'm gonna write mean instead. Average is too big of a word. Okay. So this statement simply means that as the sample mean will get closer to the true mean, so our sample mean of 100% will approach the population mean or the true mean of 50% if we increase the sample size, okay? So what I'm saying right now is 5 coin flips is not enough. This is one of the reasons that I ask you guys to do your science fair projects and do multiple tests, okay? Getting an interesting result or a really one-sided result or outcome from one test is not good enough. That is not valid. It's the scientific method. The minimum times you should test something, and I would remember this as you proceed through high school, and if you choose to go to college, you'll need to have an understanding of scientific method. Uh, Basically, the golden rule is you should always have a sample size of at least 30. At least 30 is the minimum required in order to rule out most of the confounding variables, most of the inconsistencies that you might see. So again, uh, if we get a weird sample mean, like for instance, we flip a coin 5 times to get heads 5 times, I mean, our sample average is 100% heads, we have not done enough tests, and we need to increase the number of tests or increase our sample size of coin flips in order for our sample average to get closer to the true average. So let me show you what this looks like. All right, let's say I decide to flip the coin more than 5 times. And after flipping the coin 30 times, put n equals 30. After flipping the coin 30 times, I get 17 heads and 13 tails. Okay? Which, it's not perfectly 50%, right? Well, actually, it's probably better if I don't write tails there. Let me do this instead. I'll write heads here, or H. Let's say I got 17 heads out of 30 flips, meaning the other 13 were tails. Well, that is still not exactly 50%, but it's closer to 50%, and it's approaching 50% from where I first started with the 5 heads in a row on my first few flips. Okay? So that's what this means, all right? And again, why is this important? Because you need to understand that Mendel did his experiments with these pea plants hundreds of times, probably thousands, right? And this is why we're able to reliably assign probabilities when we're working with genetics, okay? Because we understand how it works. We understand what factors are at play to contribute to the expression of specific traits, okay? So Mendelian genetics is very simple, but very concrete and nearly impossible to refute due to the basic probabilities that we're working with. Okay. All right. Let's go ahead and look at our book. All right. So how can we use probabilities to predict the outcomes of generations? Well, this goes back to our discussions about how alleles separate during the formation of gametes. So let's take a look at it right here, right? Now, each particular, well, actually, before we get there, let's start with the parents, okay? The parents in the F1 generation, each of them are tall, and each of them have a heterozygous genotype, meaning they have a dominant allele and a recessive allele, uppercase T, lowercase t, right? So, what happens is when they form gametes, they split those alleles up. Okay, they split it up into a dominant T and a recessive T, and each individual gamete that this plant makes will have either a dominant allele or a recessive allele in it. And we can reliably predict that approximately half of the sex cells that this plant produces will have a dominant T allele, and half will have a recessive T allele. Okay, and it makes no sense to assume otherwise. It's not like there's another possibility here. The only two possibilities are dominant T and recessive T. And when we split them up, we're always splitting up a pair of them. So we should always have an equal amount of each allele, right? And same thing with this other parent too. The same ratio of dominant alleles to recessive alleles will occur, right? Which means that in any particular pairing in this case, when I say pairing, I mean a combination of sperm and egg cell that leads to fertilization and subsequent reproduction. Uh, each plant has a 1/2 chance to contribute either a dominant allele or a recessive allele to the offspring's genotype. Okay, so it's possible that, let me try to use a brighter color, orange. Yeah, that's good. Let me use a thicker line. That's better. Okay, so it's possible that this parent uh, can contribute a dominant T allele to one of its offspring, right? And it's a 1/2 chance. Either it can contribute a dominant or a recessive. Let's say it contributes a dominant allele. Okay. Well, the other parent can also contribute a dominant allele, but it also has a 1/2 chance between dominant and recessive. Okay. So in order for us to determine the probability of this specific genotype, this homozygous dominant genotype, is 1/2, again, the probability of donating one dominant T, and the probability of donating another dominant T. We multiply those together, and we get 1/4, which is displayed down here in this particular diagram. Okay? So there's a 1 in 4 chance that when these two offspring reproduce and give birth to an offspring, there's a 1 in 4 chance that the genotype for the offspring will be homozygous dominant, or have two dominant T alleles. Okay, and this can be applied to any of these combinations. Okay, again, it's possible for this plant. There's a 1 in 2 chance for it to donate a dominant allele, and then there's also a 1 in 2 chance for the other plant to donate a recessive allele, and then we get this heterozygous dominant T recessive T combination or genotype. And we could also add those together, right? We could also add the probabilities together. So even though this specific combination and donation from each parent is a 1 in 4 chance, it happens twice. It's possible that the first parent can contribute a recessive allele, and the second parent can contribute a dominant allele, and then we get this same genotype, but we just receive the dominant allele from a different parent. And we could add those together. We could add those probabilities together because it's the same outcome. When you add 1/4 plus 1/4, you get 1/2. And this would be the probability of being a heterozygous tall plant, right? So there's a 1 in 2 chance, or 50% chance, that the offspring will have a heterozygous genotype with a dominant and recessive allele. All right. So that's where we're at so far. And those are the probabilities of genotypes. Again, I'm only looking at the combinations of genes. We need to go back and talk about phenotype. Okay. Phenotype. Again, whatever the express trait is our phenotype. So what are the phenotypes in this diagram that we see? Well, what are the two possible outcomes of this plant's specific characteristics? Either tall or short. How many of these plants out of 4 will be tall? Well, 3 to 4 will be tall. Very good, Bryce. And 1 out of 4 will be short. So now we know the probability of our phenotype as well. We know the we see the probability of the genotypes here, and again we're going to add these together and just say it's 1/2. There's a 1 in 4 chance of having a homozygous dominant. There's a 1 in 2 chance of having a heterozygous genotype, and there's a 1 in 4 chance of having a recessive, a homozygous recessive genotype. But we also have probabilities for how the plant will look, what trait it'll express. And we know that because tallness is dominant and shortness is recessive, that 3 of these gene combinations will result in a tall plant, which means the probability of a plant being tall in the offspring of these 2 parents will be 3 out of 4, or every 3 in every 4. And the probability of a plant being short in this generation of offspring will be 1 in 4. Okay? So that's how this works, right? Uh, pretty straightforward, and I hope you guys caught as much as you can. Um, I have to make sure that we're all at kind of an even keel. Otherwise, some of us are going to have a much more difficult time than others. Okay, so we already know what phenotype and genotype are. We know homozygous, heterozygous. We've understood how probability can be used to calculate specific outcomes in genotypes and in phenotypes. Okay, let's go ahead and talk about how we can put this in a very simple diagram. And that diagram is a Punnett square. Okay, so you guys are probably looking at this like, oh, Mr. Holden, I don't want to draw these stupid diagrams to calculate probability. Look at how much work this is. This sucks. Screw you. I'm not doing it. Well, I counter that by saying it's possible to do it in a very easy box called a Punnett square. And that's what we're going to be looking at right now. Okay? Let me erase all this nonsense. Yikes, that's a lot. How do I reset all the notes? There's a way to reset them. No? 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 How do I reset notes? Let me look this up on Google. To reset notes, Microsoft Edge. Right click the start menu. No. I'm just gonna erase them. Screw it. Alright. Zoom in. I drew too much here. My bad, guys. I should have erased it earlier. Why couldn't one of my students help me erase my board? Wave the mouse. Wave the mouse. Alright, I think that's good. Yeah, we're good. Okay. I don't think there's no undo on this. There's no undo on a PDF document. So I gotta figure out how to do it a little bit easier. What's this? Touch writing? I don't know what that is. Alright, so Punnett squares. What is a Punnett square, and how do we use it to calculate probability of genotypes and phenotypes? Well, let me show you this right now. So, here we have a couple of parents' genotypes. So they're both heterozygous with a dominant B and a lowercase b. I don't know what this particular allele, what trait it controls, but let's just let's just assume that whatever it does control, the uppercase B is the dominant allele, right? Okay, so again, remember that the alleles separate during the formation of gametes. So once we draw this square, we're no longer going to be considering the parents' genotypes as being together. Okay, we're going to separate them out, and we're going to write them like this. Oops. So we have a box, right? We just draw a box like this, and then we draw a giant plus sign through the middle of it. And that's how we get our Punnett square, okay? Oops, that was disgusting. Okay, so here we are. Is this brown eyes and blue eyes? I don't think so. This is something for rabbits. I don't know what particular gene it is. Okay, So how do we proceed from here? And let me change the size of my note taker thing. Uh, well, there's a couple rules I want you guys to follow when you're doing this. First off, is that every single allele that you see on the top, like right here, you're going to be writing those in every square that happens below it. Okay, so it doesn't matter if I have just two alleles here, if I expand this to have four alleles, like a two-factor cross, or even six alleles in a three-factor cross. I mean, it doesn't matter what it is. All of the alleles on top, you're going to fill them in each box below. Uh, By contrast, the alleles that you see on the left side of the Punnett square are going to always be filled into the boxes on the left. Sorry, on the right. Okay, every single box to the right afterwards, you're going to write in those alleles. Doesn't matter how big the square is, you're just going to keep going until each one has that particular allele. How do you know which allele to write? Well, they're lined up. Okay, since B is lined up with these boxes, I know I'm supposed to write it here, and I'm supposed to write it here. And I know this recessive B is lined up with these squares, so I'm writing in that square and that square. And same thing for the ones on top. It's just going to be writing it in all the squares below it. You're not going to write it across like this. Actually, there's a little too much here. You do not do this. You do not write the allele across like that. That is a big no-no. Don't do that, okay? Instead, you're going to go all the way down in a straight line. Don't do that diagonally crisscrossy nonsense, okay? It's going to make me very upset. I'm going to have a very tough time, all right? All right. So another rule is I like to follow it because if you don't follow it, it makes my eyes hurt trying to see if you got the correct answers. And I don't want my eyes to hurt because I'm already looking at a computer screen all the time. And you know, just take it easy on poor old Mr. Holden. Okay? So here's the second rule. Do the dominant alleles first. Always do the dominant alleles first. Don't start with the recessive alleles, okay? Don't start with those first. Do the dominant ones first, and there's a reason you do the dominant ones first, okay? So let's go ahead and start off by doing the dominant alleles. So I'm going to start with the ones on top. You can start with the ones on the side or the ones on top. It doesn't matter. I like to start on top. That's just me. So I have a dominant B right here, and I'm going to simply write it into all the boxes below. And when I write it, I'm going to start from the left side of the box to give myself room for when I write my next allele. Okay? So I'm always going to start writing. I'm going to always write in the alleles from left to right. Okay? And then as I have to write in more alleles, I'll just keep including them after whatever allele is already there. So I put a dominant here. And again, I'm doing the ones on top. And then I put a dominant B here. Now I could do the dominant on the side. Okay? Since there's already an allele in this particular square, I'm just going to write the next allele right beside it. Okay? And then I also have to include one here. Boom. So now I filled in all of the dominant alleles in this square. Now I can do the recessive alleles. So I'll switch up the colors for that. Let's do blue. So now I'm going to start on top with the recessive B. And I'm just going to fill it in every single box below. Okay? And again, if there's already an allele in that box, I'm just going to write that new allele right beside it. And now I have to do the recessive B on the side. I'm going to write it across. Boom. Boom. And there you go. That is what your genotypes in your square should look like. Let's break this down for a second. So, what we're looking at is essentially the same outcomes that we got in our diagram earlier. So we have one out of 4 chance to be homozygous dominant. We have a 1 out of 4 chance here, a 1 out of 4 chance here, and a 1 out of 4 chance for homozygous recessive. Again, we're just going to consider the heterozygous genotype to be 1 out of 2. Okay? We're going to add them together. So now we can determine our probabilities by simply counting how many boxes out of 4 have a specific genotype. So for homozygous dominant, our probability is, again, 1 in 4, because only one box out of the 4 that I have has this specific genotype. For heterozygous, well, I have 2 boxes out of the 4 that have that specific genotype. And then again, I only have 1 out of 4 boxes with the recessive, homozygous recessive genotype. Okay? So again, this is how we calculate probability using this square. And you don't have to do any math. You don't have to do any division or multiplication at all. Oh, you have to do addition, actually, if you're adding up these. So forgive me. You do have to do some math. But it's simple math, all right? It's not too difficult. Okay, so why do I make you guys write the dominant alleles first? Well, this is why. Mr. Holden hates this. Mr. Holden hates this with his life. Every time Mr. Holden sees this particular version of the heterozygous genotype, it makes Mr. Holden very angry, okay? Because you should never write the recessive allele first, right? It just makes sense. The dominant allele is the one that's expressed over the recessive allele, so why should you write the recessive allele first? It's complete nonsense, okay? This makes me very angry. Very, very angry. So, instead of doing it like that, always write the dominant allele first so that your genotypes will always end up with the recessives coming after the dominants, and you don't get some nonsense like this. I hate this, right? Because then what happens? You go to write your probabilities for genotypes, and then you put something like this, and I'm just like, yo, these are the same thing, doofus. Write them together. So yeah, again, don't write your heterozygous genotypes like this. It makes me very upset, and it hurts my eyes. There's nothing else but the 11.2 worksheet, so you guys are good. Should we end class now, or should we talk? Oh wait, hold on a second. Hold on a second. Let's go ahead and do the phenotype probability. So again, I don't know what trait this allele codes for, but someone tell me, give me an example of what these 2 alleles could code for, or what versions of a trait that they could be for a rabbit. Sammy, you want to help me out here? Sammy, give me a possible trait for this uppercase dominant B. You're not helping, Sammy. Okay, let's say that big teeth. Okay, big teeth. So a rabbit that has the dominant B allele will have super big teeth, and then lowercase will be small teeth or big ears. Okay, that's fine. This is weird. So because big ears is recessive, right? The only time it will be expressed is when there is no dominant allele present, right? So, what is the probability of having big teeth in this particular cross? Remember, we're only looking at the genotypes that are inside the boxes, okay? Everything inside the box or inside these squares are the offspring genotypes. So what's the probability of having big teeth in the offspring? We're not worried about the parents. The parents are on the outside. It doesn't necessarily have to be little. Recessive alleles are not necessarily little or lesser versions of a trait. So someone tell me the probability. We're not looking, Natalia, we're not looking at genotype. We're looking at phenotype. I don't care what the genotype is when I'm talking about phenotype. Okay, what is the probability of having big teeth? Okay, so hold on. We already have those genotypes, Natalia. We already have them right here. You're just being redundant. I already know that it's a 1/4 chance for 2 dominant Bs, a 50% chance for dominant recessive, and a 1 in 4 chance for recessive recessive. Guys, the probability for having big teeth in this generation is 3 out of 4. Okay? Remember, this is going to result in... Oh, I can't even write that there. This offspring will have big teeth because big teeth is dominant and it only has the allele for big teeth. Okay? This offspring will have big teeth because again, big teeth is dominant, right? Doesn't matter that we have a big ears allele there because big teeth is dominant, it will be expressed. Same thing with this particular offspring. This offspring also has a dominant big teeth allele that is overpowering the recessive big ears allele. Okay? And then this individual only has two recessive big ear alleles. So of these four offspring, three of them will have big teeth. That's what I'm looking. That's what I mean when I say the phenotype probability. I don't care what the genotype is. I already see that based on me filling out this Punnett square. Okay, so the big ears. How many out of four will have big ears? 1 in 4. Okay, 1 in 4. So we see that the probability of genotypes is not necessarily the same as the probability of phenotypes. And again, this is genotype. I'm going to put geno here. But what I discussed over here is phenotype. And I'm not... The only way I care about genotype is because it might tell me what phenotype I'll have. All right? But I'm not going to just assume that uh, the genotypes for big teeth should be separated in my phenotype. Both the homozygous dominant and the heterozygous genotypes will give me big teeth. Okay, I don't need to separate them. Understood? So you guys are doing great when it comes to the genotype probability, but you guys got to remember phenotype is different. It's just whatever the characteristic is in that offspring. Okay. There are only two possibilities here, big teeth or big ears, right? Whereas in genotype, again, we have three possibilities. All right? Connor, I know you have to go right now, so that's fine. What I'm going to be covering next is not part of your homework, but it will be part of our exam, and it will be something that you guys have to complete in order to earn credits on your next test. So I'm going to erase all this stuff. And we're going to go to another cross. Get out of here. Yuck. Ugh. Takes me forever to erase this stuff. All right. So, well, before I go to that, this is what we call a one-factor cross because it only shows one gene, or essentially one gene. Uh, there's only, although we have two alleles for every gene, we still consider that combination to be one gene, right? We only look at one pair of alleles. Think about it that way. One factor cross, you can consider it a one pair cross. A pair being a pair of alleles. Okay. So in this instance, our Punnett square will look a lot simpler and it'll be a lot easier to complete. But since not every single trait out there is controlled by a single gene, we need to draw more complex and larger Punnett squares when we're dealing with traits that are controlled by multiple genes. So in some instances, and you will have to do this during our unit, you will have to complete a two-factor cross, or in this case, a two-pair cross, meaning we have two pairs of alleles, right? And this happens quite often because again, most genes are controlled. Oh, sorry, most traits can be controlled by more than one gene. We see that the more complex the animal is. A pea plant's rather simple. Humans and rabbits are a fair bit more complex, so we have to rely on these larger crosses in order to complete them. But the same rules apply in the large cross as they do in the short cross. The same rules apply in the two-factor cross as they do in the one-factor cross. Okay, which means I'm going to start with my dominant alleles first, and I'm going to fill all the dominant alleles in, and then I'm going to fill in the recessive alleles. The difference is one extra rule is that I'm only going to do one specific allele at a time, right? Meaning I'm only going to do the T's first. or sorry, I'm only going to focus on one gene at a time. So I'm going to focus on the T alleles first, and then I'll do the G alleles afterwards, okay? So I'll just do them one after the other, and that way all of my genotypes in these squares will come out really neat in the correct order, and the T's and G's won't be mixed up, okay? Now, when I look down at what the book does is good, actually. I'm looking at this and it looks solid. None of the problems. All right, good, good, good. Okay, you did well, book. Thank you. So you notice that when they complete the cross for you, they actually put the dominant alleles before the recessive alleles in each one. Okay. So let's go ahead and do this. Let me show you guys how this works. Yes, Bryce. Yes, Sammy. You can go to the bathroom. Go ahead. You do dominant first. So since we're going to start with T's because T is the first allele written here. If it was GT instead of TG, I would do G's first. But because T is written first, I'm going to do the T gene. Okay. So I'm looking at the dominants. Here's the dominants. There we go. And I'm just going to start filling them in. I'll start with the top first, and I fill them in each square below. Just working my way down like this. Okay. Now I do the ones on the side, so I'm going to write these ones across. And again, if there's already an allele there, I just write the one I'm doing right next to it. Oh, whoops. I made a mistake. Not supposed to be there. It's supposed to be right here. Okay. So far, so good. And now we can do the recessive alleles. So let me erase the stuff for the dominant alleles. I've already done those. Now I can move on to the recessive alleles. There's the two right here, and there's two over here. So I'm going to start with the ones on top first, and I'm writing them in each square below. And then I'm going to do this one. And now I'm going to do the ones on the side. Make my way across. By the time I'm done with all of the T alleles, each square should have 2 T alleles in it. I'll just double check to make sure. Yeah, each of them have 2 T alleles in the square, so I'm good. I can move on to G. Alright, erase this. Let's do G now. Start with the dominants first, and away we go. I'm going to go up for the ones on top and write them below. This is going to be tricky. Running out of space. Sucks writing with a mouse. Okay, now I'm going to do the ones to the side. And this one. Okay, now that I'm done with the dominants, I can do my recessives. Yep. Recessives are here and here. I'm going to start with the ones on top first, filling them in below. Making my way downtown. Okay, now I'm going to do the ones on the side, filling them into the right. It takes some time, yeah, that's for sure, but if you do it this way, you'll get it right every single time. Provided you don't, make any careless mistakes like I almost did earlier. So, now that I'm done, each of these should have 4 alleles, and none of the genotypes should have the recessive coming before the dominant, and it looks like I'm good. Yep, this is a perfectly written 2-factor cross. Natalia? I did the dominant T's first. I did them first. I could start over again. Natalia, I know this looks complex, but let's go back to the simpler one before we go back to this one, okay? Again, I always start with the dominant alleles first. The reason I said I started with T first over on the 2-factor cross is, again, because T was written first. If it was GT instead of TG, I would do the G's first for all of these. But in every single one of these, the T gene comes before the G gene. And then I just work my way. I'm not going to redo this cross, but I'll do this one again. Okay. Uh, I'm going to start with the dominant alleles first. So here are my dominant B's. The ones on top, I'm writing in the squares below. The ones on the side, I'm writing in the squares to the right. So this one is going to go here. And it's also going to get written down here. This dominant B is going to be written in every single box to the right of it. Oops. So I'm going to write one here, and I'm going to write one in here. Oh no, Natalia! Natalia, how could you fall asleep? This lesson is so fun. All right. Let's do this one more time again. Let's do this one more time again. I'll try to make it fast this time. But Natalia, please stay awake for this, because this took me a while and my hand's starting to hurt. Okay? I need to get like one of those little writing tablets for the computer. Those are probably pretty sick. I need to tech out my entire thing. Did I do one wrong? I don't think I did one wrong, Bryce. Did I? Let's see. I can't check because I can't undo. I don't think. Now I really want to know what I did. Dang it, Bryce! You're sowing discord and doubt in my mind. All right, Natalia, are you listening, Natalia? Bueller. I have 10 minutes to finish this. Okay, I want to make sure we're good. So, again, I'm following the rules that I established in that other cross, which means I'm going to do my dominant alleles first. Okay? And the reason I'm doing T first is because it's written before the G, right? If it was GT, I would do G first. But I'm doing T. All right? Now, I'm going to simply look at where my dominant T's are and fill them in the corresponding boxes. So again, the ones on top, I'm going to write them below. Okay? The ones on the side, I'm going to fill them into the right. All right? So here we go. I'm starting with this one first. Starting with this particular dominant T first. And I'm filling it in every single box below. Here's one. There's the next box, next box, next box. I'm done. Okay? I'm just going to go ahead and cross it out. Don't need to worry about it. Now I need to do the next one. This particular dominant T, I need to fill it in every single square below. I'm done with it. I'm crossing it out. Now I got to do the dominant T's on the side. So I'm going to fill them into the right. This one's going to go in every single box to the right. Done. Crossing it off. Now I'm going to do this dominant T. Done. I'm going to cross it off. Okay? Now I have to do the recessive T's. Natalia, the rule is I'm going to do one gene at a time, and I'm going to do the dominant alleles for that gene first. Okay? One gene at a time. In this case, I'm doing the T gene, which means either the dominant or recessive T's, and then I'm going to do the G genes. Okay, the G alleles. Right? Make sense? Hopefully. Oh, I did make a mistake. Oopsies. Okay, it happens. All right, so now I'm going to do the recessives. I'm going to start with the top ones first, so I'll do these two here, and then the ones on the side afterwards. Okay, so I'm going to fill this recessive T in every single box below it. Cross it off. Do the next one. Every single box below is going to have this recessive T. Cross it off. Let's do the one on the side. Every single box to the right of this recessive T should have another recessive T written in. Boom. Cross it off. And the next one, here's our last recessive T. Again, writing in every single box to the right. checking to make sure that each of my squares has two T alleles in it. Each of them do, so I can move on to the G's. And I'll do the G's now in red. Again, I'm going to start with the dominant G's first, and then move on to the recessive G's. Okay? No, not all. Again, I'm going to do I'm going to do one gene at a time, meaning either the T gene or the G gene. And then I'm going to do the dominant alleles in that gene first, and then the recessive alleles. Okay? I'm sorry, I should have explained that a little more with a little more clarity earlier. You do. Oh, okay. Well, thanks, Natalia. I'm going to finish these again. All right. So dominant G's first before I do the recessive G's. Again, I'm filling the dominant G's below the one at the top here. And then this one. And now I'm done with this one. I'm done with this one. Let's go ahead and do the ones on the side. So this particular dominant G will get filled in to the right. Cross it off. And now this dominant G. Cross it off. Okay, the only thing that's left is my recessive alleles, my recessive G's. Let's go ahead and get them. I'll start with the ones on the top, filling them in below. Recessive, 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 and recessive, 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 recessive. All right, now I'm going to do the ones on the side. Okay, filling them in. Oh, let me cross these out. These are done. Recessive, 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 cross it off. Recessive, 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 and recessive. Cross it off. There will never be more than 4 in a square. Never be more than 4 in a square. Okay? So this one, I'm pretty sure I did this one accurately. I'm pretty sure I didn't make any mistakes. But Natalia, do you feel more confident? You said you know how to do it, so I'm assuming that you do feel more confident. You're not going to be doing this on your homework tonight, so don't worry about it. I fixed the mistake. Good. Thank you, Bryce. All right, guys. So let me finish erasing this, and I'll show you guys the Google Classroom page. It's taking a while. All right, we're good. Okay. So again, your homework is this 11.2 worksheet, and the reason I make it a PDF, Natalia, is because if I make it a Word document and you try to open it with Google Drive, it's just going to look like nonsense. Okay, but you can I'm pretty sure you can convert it to a Word document if you so chose to. It's just that for me, I want to make sure it appears the way that it would appear if I printed it out. So that's why I use the PDF. Sorry, it makes it inconvenient for some people, but for me, I just think it's a more consistent way to share the file. So here it is, due on Friday. That will end our class. You did it, but it was all jumbled. You can print and scan it back. Of course you can. Natalia, it's just gonna take some practice. I still make mistakes, and Bryce caught one of my mistakes. So, and I've been doing this for years. So it takes a while, and you got to be very careful. Oh, you did it, but it was all jumbled. Oh. All right, Natalia. I thought you were talking about the Punnett squares. But if your work is all jumbled, that's fine too. I'll sort it out. Rip my eyes. Natalia, you know what's interesting? Is that... Here, I'll show you. When you typed out I don't want to print it, I heard a voice in my head, and the voice sounded like this. Okay, it sounded like, "But I didn't want to print it." That's what it sounded like. You don't have to print it. If you do it the way you have to do it, that's fine with me. All right, Bryce. Bye. Take care, everybody. We're done with class for today. Geez, we took way longer than third period, but I think I explained things a little bit better to you. They were kind of like jaded by the time We were like halfway through class. Alrighty, everybody. Wait, attendance has been an issue. Let's see who is here. I know Christian, Natalia, Adam, Bryce, Sammy. I'll just do an attendance check on the classroom page. So before you guys leave, or you check in your other classes, stop by and do an attendance. Just respond to my attendance check here. All right, and that'll be all. Everybody, take care. Have a good rest of the day. I will see you tomorrow.
I feel your eyes in the morning sun. I feel you touch me in the pouring rain. And the moment that you wander far from me, I wanna feel you in my arms again. And you come to me on a summer breeze, keep me warm in your love, then you softly leave. Well, it's me you need to show. How deep is your love, your love? How deep is your love? I really need to learn, 'cause we're living in a world of fools, breaking us down when they all should let us be. We belong to you and me. I believe in you. You know the door to my very soul. You're the light in my deepest, darkest hour. You're my savior when I fall. And you may not think that I care for you, when you know down inside that I really do. Well, it's me you need to show. How deep is your love, your love? How deep is your love? I really need to learn, 'cause we're living in a world of fools, breaking us down when they all should let us be. We belong to you and me. Then you come to me on a summer breeze, keep me warm in your love, then you softly leave. Well, it's me you need to show. How deep is your love, your love? How deep is your love? I really need to learn, 'cause we're living in a world of fools, breaking us down when they all should let us be. They belong to you and me.
I was born by the river in a little tent. And oh, just like the river, I've been running ever since. It's been a long It's been too hard living, but I'm afraid to die. I don't know what's up there beyond the sky. It's been a long time coming, but I know change gon' come. Oh, yes it is. See, I go to the movies and I go downtown, but they tell me they don't want me to hang around. It's been a long time coming, but I know a change gon' come. Oh, yes it is.
Angels gathered in their masses, just like witches at black masses. Evil minds that plot destruction, sorcerer of death's construction. In the fields the bodies burning, as the war machine keeps turning. Death and hatred to mankind,